Good afternoon. So I'm going to be presenting on the paper Adaptive Plasticity of Vaginal Innervation in Term Pregnant Rats. So the female reproductive system undergoes major structural and functional changes, which includes alterations in receptors, alterations in endometrial and muscular size, as well as immune function. These changes are important in ovulation, sperm transport, implantation, gestation, and parturition. Pregnancy is accompanied by massive degeneration of sympathetic axons. Reproduction related plasticity is not defined to target tissues, however. Studies show changes in reproductive status, which also induces reductions in uterine sympathetic nerves. And the photo included in the slide is the basic reproductive system of a rat model. So to continue, as stated in the paper and hypothesized by the researchers, the increase in estrogen equals the depletion of uterine synthetic fibers, which is also related to the axonal degradation as pregnancy is accompanied by. It is thought that reduced excitatory innervation may be important in preventing neurally mediated uterine contractions at stages of the reproductive process. So throughout the paper, the term innervation is included. I do have the definition of peripheral innervation, which is also explained in the paper, which is the scheme tracing innervation of body regions to the peripheral nerves, which receives motor and sensory nerve supply. So anytime innervation is brought up within the paper, that is what it refers to. So the rodent vagina contains abundant sensory and autonomic innervation, which is thought to be important in regulating vaginal smooth muscle tone, as well as glandular secretions, blood flow, and sensitivity. The term surgical menopause described is induced by bilateral ovarectomy results and increased densities of vaginal sympathetic, parasympathetic, and sensory axons relative to intact cycling rights. Rats, not rights. Rats do have rights, though. So the question asked in the paper is, do heightened levels of estrogen in late-term pregnancy cause parturition? Parturition is also the loss of pregnancy or the loss of the fetus, or miscarriage, as commonly termed to our language. So 20 Sprague-Dowley rats at about 60 days of age were used for this experiment. So there were daily vaginal smears collected, 5 tissues collected at estrus, which is the reoccurring period of sexual receptivity and fertility, 5 at diestrus, which is the period of time between estrus where there is inactive sexual activity, and 10 from proestrus, which are already mated females and already pregnant. And these embryos are at E10 and E21. E21 is about term for rats. Vaginal tissues from all rats were sectioned and fixed in H&E staining. And then those were described under the fluorescent confocal imaging of the vaginal cells. And that determines submucosal tissue and innervation density, which is a means for each marker compared by estrus and diestrus. And again, below is the reproductive system of the female rat. I have done some research, and the lab that I currently work for, a lot of it is on various embryonic stages of the rat. It doesn't really have anything to do with this paper, but just this is a good basic anatomy showing where the ovary, oviduct, the urinary bladder is, and the uterus, which you can see is right along here. And obviously, with amounts of pregnancies that these rats will have, the uterus will grow, will expand and contract along with the babies after they are born. So figure 1 on this paper There's several images. So they are the immunofluorescent photomicrographs of the vaginal innervation for diastrus and A through E and estrus F through J. These sections were stained and they served as a marker for sensory nerves. So here are several images, several days, day 10, and then the corresponding letters for either diestrus or estrus in the vaginal smears. Figures 2 compare the axon density percentage along with the specimens that were stained. Which you see we have PGP, TH, tyrosine hydroxylase, and vesicular acetylcholine transporter, calcitonin gene-related peptide, and transient receptor potential vanilloid type 1, which does sound a lot like vanilla. And now I'm hungry. Anyways, so these figures are the quantitative analyses of the central vaginal innervation in the estrus cycle. And these immunoreductive fibers were expressed as the percentage of the total field area on cycle A, or the graph A here. And then graph B, these values were normalized for changes in target size by multiplying the percentage area by the total area of the submucosal compartment. And then these data were obtained during diastrus. The black bars are diastrus, and then the open bars, the white, are estrus stage. So you can see in the comparison of the axon density during the diestrus versus estrus. So this resulted in fiber densities that are lower in estrus, and all the distributions were similar. However, there were significant reductions occurring late in the term E21 pregnancy. So what does this mean? This means that as far as diastrus and estrus goes in the vaginal innervations, there are similarities between these two, and they're closely related in their tissues and responses to circulating hormones. The uterine epithelium shows marked morphological changes along with the estrus cycle, and the myometrial smooth muscle mass varies across the estrus cycle stages, but the smooth muscle size increases at proestrus, breeding, and estrus relative to diestrus. There are differences that exist in tissue responsivity to hormones, and perhaps there are other factors with respect to morphological plasticity. However, there is no significant change demonstrated in the overall axonal area or in any of the subpopulations. Estrogen also increases the thickness of vaginal muscularis. This hormone is likely to be a factor in promoting the increased size of muscular and submucosal layer in the latter stages of pregnancy. However, the loss of sensory innervation could serve a major adaptive role with regard to pain sensation during birth, which makes a lot of sense as to why in humans, there's some women I know that talk about pain threshold for birth not being as bad. Maybe there are differences in the sensory tolerances of women, which could be a further study to look at. And then to conclude, the term pregnancy, at least in rodents, is characterized by significant reductions in the innervation of the vaginal canal. So here's a YouTube video that I posted, which is kind of going into like the broad spectrum of rat breeding. It's pretty long. I wasn't going to add it along with this video because I believe it's like 19 minutes long. It's really interesting. I do a lot of breeding work at Yale, so I thought this video kind of went pretty well with this paper. ways. Thank you very much.